So next up is Zhixun Dou, who's from Mass General and Harvard, and he's going to tell us about nuclear autophagy in senescence and aging. All right, looks good.
Let me unmute myself.
Now you're out of the slideshow mode. I don't know if that was intentional.
Okay.
There you go.
Yeah. Can you hear me all right? Okay, great.
Yeah.
All right. Thank you, everyone, for coming here. And I too want to thank the organizers for putting everything together. So I go by Dou. Dou is my last name, but apparently it's much easier to pronounce than my first name, Zhixun, so I just go by Dou. It's much easier. So I will talk about nuclear autophagy in senescence and aging. So I'll start by talking about cellular senescence, which is an important hallmark of aging. And then I will also talk about other hallmarks related to senescence, for example, how it contributes to genomic instability, epigenetics, and the loss of proteostasis. So I'll first talk about the cellular senescence. And we all know that cellular senescence is a stable form of cell cycle arrest. Right? So this is permanent shutting down of the cell cycle. And then from the normal to senescence, a lot of things happen in the cells. For example, you see the cells getting bigger, and then they are more enlarged and flattened, and then the nuclei also look different. So this is a very stable form of cell cycle arrest in comparison to the quiescence, in which the cells can still re-enter the cell cycle. But senescence, by definition, is a permanent loss of cell cycle. Now, a lot of things can induce the normal to senescence transition. For example, the cells can experience telomere attrition, as was originally discovered by Hayflick. In addition to the replicative senescence, other acute stress can also induce the normal to senescence transition, for example, in response to DNA damage or activated oncogene expression. Now, this induction of senescence can be beneficial. For example, when the cells are experienced activated oncogene, the cells can turn on this stable form of cell cycle arrest program, and this is a potent tumor suppressive mechanism to shut down the cell cycle. In addition, senescent cells secrete a lot of pro-inflammatory cytokine. This will recruit the immune cells, leading to immune-mediated clearance. So there's certainly a beneficial role of senescence. Now, on the other hand, in the context of aging, we know there are a lot of senescent cells that are accumulated in the tissue, and then the accumulation of senescent cells will block the cell renewal and the regenerative capacity of the tissue. And also, the senescent cells secrete a lot of inflammatory cytokine, the so-called SAFs, and then this will cause chronic inflammation that contributes and promotes most of the age-associated diseases. So that's why, given the important role of senescence in cancer aging, there's a lot of interest to target the senescent cells, either by killing them, the so-called senolytics, or using drugs to block the inflammatory phenotype, the so-called the xenomorphics. Now, as I mentioned to you, so a lot of the things are happening in the senescent cells from the nucleus. So this is what a normal mammalian nucleus look like. You have the nuclear pore complex, and you have the double membrane nuclear periphery, and then there's this nuclear lamina meshwork beneath the nuclear envelope. And so these are the type 3 cytoskeleton. They provide the nucleus with mechanical strength, and then they also regulate the higher chromatin organization. And so we know a lot of the chromatin is anchored to the nuclear periphery through the nuclear lamina, and these regions are repressed for their gene expression. So a lot of things happened during this process in senescence. For example, one of the most prominent changes is the loss of nuclear lamina, specifically the lamin B1 isoform, and this will lead to the chromatin deassociated from the nuclear periphery, and then a lot of the chromatin organized into a heterochromatin foci. And then there are other changes for the nuclear envelope as well as the nuclear pore complex. So as I mentioned, one key hallmark of senescence is the specific loss of the lamin B1 isoform. And we have shown that the loss of lamin B1 is happening at both transcription and the protein level. and the protein level is mediated by degradation by autophagy. So this is an example from primary human fibroblasts, and you show in the control cells that lamin B1 is nicely localized at the nuclear periphery. And when you induce senescence by multiple means, what you see is that the overall intensity of lamin B1 is reduced, and you also start to see the cytosolic structure that is also positive for DAPI and for specific heterochromatin marks. And so Peter Adams' lab originally discovered the cytosolic chromatin structures, and then we collaborated with his lab by showing that the cytosolic structure can actually be targeted by autophagy for degradation. So in the next slide, I'll briefly introduce what is autophagy and what is this molecule LC3. So autophagy is a catabolic membrane trafficking process, or called self-eating, that's initiated with the formation of isolation membrane, and that later on forms the double membrane autophagosome, and that later on fuse with lysosome, and the content is degraded by autolysosome. Now, this molecule LC3 is a very important molecule involved in the autophagy process. So LC3 in the normal condition is a diffuse pattern, but in the setting of autophagy, so it's recruited to both outer and inner membrane of the autophagosome membrane. So the inner membrane pool of LC3 will recruit the substrate, so it directly binds or indirectly binds to the substrate and send the substrate to the autophagosome for degradation. Now, upstream of LC3, there are a lot of autophagy genes regulating this process, for example, ATG5, ATG7, and among others, and then they respond to starvation and specific cellular stress. Now, this degradation process can be fairly non-specific. For example, in the context of starvation, the cells are trying to grab whatever they can eat to get through the hardship. So this is considered bulk autophagy or non-selective. But on the contrary, autophagy can be fairly specific. For example, in the process with senescence, the cells use autophagy to specifically degrade specific substrate for a particular function. So what is really weird about this autophagy adapter molecule LC3 is that in the basal state, in the control sense, right? So you see a lot of LC3 is in the nucleus, and then in the starvation or stress condition, the nuclear pool becomes empty, and then the cells form this autophagic puncta that has LC3 on it. So this has been very confusing to the field. What is LC3 doing in the nucleus? And so we were one of the first to show that there was a nuclear role for autophagy, and especially for the LC3. So what happened is that part of the LC3 in the basal state is in the nucleus, as I show you, and then a portion of the LC3 directly binds to the lamin B1 isoform and chip at the lamin-associated domains at the heterochromatin. So this is what happened in the basal state. Now, in the process of senescence and aging, what happened is that the nuclear membrane forms the bleb, and then these structures can also butt off to the cytosol to fall off to become cytoplasmic chromatin fragment that has LC3 with lamin B1 and part of the chromatin. So they can undergo autophagy degradation, and then we have shown that together with Peter Adams' lab, and this is a major mechanism to lose the lamin B1 in senescent cells as the protein level degradation. So in addition, the cytosolic DNA is sensed by the innate immunity cytosolic DNA sensing pathway, CGAS-STING pathway, that contributes to the senescence-associated phenotype. So we do think the autophagy, especially the nuclear autophagy, has a very important role in senescent cells in altering the nuclear and chromatin structure, leading to gene expression changes. So these studies are all published. So for the rest of my talk, I'm going to talk about some unpublished work from my lab, asking the question, does autophagy degrade any other nuclear substrate in senescence? Right, so I've shown you the lamin B1 as an example. Usually, if you see one, there's got to be more. So we asked a very broad question, are there any other novel substrate of nuclear autophagy in senescence? So we collaborated with Hopper's lab in Harvard to take the unbiased proteomic approach to find novel substrate of autophagy in the nucleus from the senescent cells. So our approach is that we first ask what is the nuclear LC3 interactome, what binds to LC3 in the nucleus, and then we also ask So what is specifically lost in the senescent nuclei? And we're comparing the autophagy competent versus autophagy deficient nuclei. So the overlapping pool is what we are most excited about, right? So that has the highest confidence of the putative novel substrate of autophagy. So what did we find? So the top hit is lamina B1, which is good. That serves the positive control. So the other target we found is a weird protein called WSTF. So this protein is never implicated in autophagy or senescence. So I'm going to tell you what it does and what is the biological function for WSTF in senescence. Okay, so WSTF is in fact part of the ISY chromatin remodeling complex. Right? So we know that normally the DNA is organized into nucleosome, which repress the DNA expression and transcription. And that there are chromatin remodeling complex that can open the nucleosome to expose the DNA, so activating the transcription or replication. And there are also complex that doing the opposite, that can switch the open chromatin back to closed chromatin. Now, ISY complex is a repressive chromatin remodeling complex. So it uses ATP and shift the open chromatin back to the closed chromatin, thereby repressing gene expression. Right? So ISY complex is a repressive complex to repress gene expression. So the ISY complex in mammals has a lot of different subunits. The catalytic enzyme, the ATP is the SNF2H. And then this WSTF is a particular regulatory subunit that directly binds to SNF2H. So what is really interesting is that from our proteomics data, so you only see the specific loss of WSTF in the complex, and you do not see loss of SNF2H or other complex. So indicating the cells has a very clear idea what it's trying to do. So first thing first, let me show you some validation. And we have shown that the loss of WSTF at the protein level is a common signature for cellular senescence. For example, this is IMR90 from a human fibroblast. You see when the cells are senescent, they specifically lose lamina, the WSTF, and then the catalytic subunit, the SNF2H, remain largely unchanged. This is also true in mouse and cancer cells undergoing the therapy-induced senescence. And what is also interesting is that if you look at the mRNA level, WSTF is completely unchanged. So we know this is a very specific protein level degradation. Okay, so obvious question is autophagy really involved in WSTF loss in the senescent cells? So we are answering this question using both genetic and pharmacological approaches. So first, we stably knock down the autophagy essential gene ATG7. As you see, the knockdown of ATG7 block the LC3 lipidation, so the cells are really autophagy deficient. And then what you see is, interestingly, in the control cells, when you induce senescence of the fibroblast, you'll have a very nice drop of the WSTF at the protein level. And then when you don't have ATG7, the protein level largely stay there. So we can also block autophagy using drugs. So now we induce senescence, and then you see a very nice loss of WSTF. And on top of that, we can treat the cells with the lysosomal inhibitor bufalomycin, and then you see the WSTF protein is largely reversed. Now, by contrast, if you treat the senescent cells with the proteasome inhibitor MG132, you see nothing happen, WSTF is not reversed. So we are very clear that this loss of WSTF is specifically driven by autophagy in senescent cells. Next, we are going to the mechanistic insight, how autophagy and specifically nuclear autophagy degrade the WSTF. So I mentioned to you about LC3, and in fact, LC3 is a family, is a member of the ATGA family proteins. So there are 6 members, and then they recognize the different substrate under different conditions. So the 6 isoforms give the cells the complexity and the diversity to recognize the substrate. So we have done purified in vitro experiment in which we in vitro translated WSTF and used bacteria podium. And then what you see is that the GABA-RAB isoform of the ATGA is the predominant isoform that potently binds to the WSTF. So we have further mapped the WSTF and the GABA-RAB interaction in great molecular details, and that we know the specific residues on both ends that mediate the interaction. So on the WSTF side, that this linker region, and specifically the leucine residue, is really important in mediating the GABA-RAB interaction. Right? So when we truncate this short linker or we mutate this leucine to alanine, we almost completely lose the interaction with GABA-RAB. So the obvious question is now, what is the behavior of this mutant in senescent cells? Is it still being degraded by autophagy? Right. So now we stably express either the wild-type WSTF or the two mutant that doesn't bind to GABA-RAB. And then what you see is that in the control senescent cells, the WSTF is nicely reduced by autophagy degradation, and then the mutant that doesn't bind to GABA-RAB largely stay there, indicating the binding to the autophagy adapter is really important. And we also see that in the wild-type WSTF, you see the nucleus of cytosol trafficking followed by cytosolic autophagy, lysosome degradation. And these two mutants largely retain in the nucleus, so they have trafficking problems. Okay, so after all that, so what does WSTF do in senescent cells? Why do the cells really want to degrade specifically the WSTF subunit of the ISY complex? Is there any biological significance to this? Right, so now, to do this, we counteract with what the cells are trying to do. So if you want to degrade it, we force to overexpress it. And then we ask the question, what happened to the cells? All right, so you see in the control cells, the WSTF is nicely lost. And we use a lentiviral vector to stably express WSTF. And we ask what happened to the cell. You see the loss of lamina P1 or the induction of P16 as senescence markers are not really affected. But what is interesting is that in the control cells, you see a nice induction of L8, which is a marker for the senescence-associated secretory phenotype. And then when you put WSTF back, the L8 expression is largely repressed. So now we can also do this at the genome-wide level to look at the RNA-seq. And then what you see is that in the control senescent cells, mediated by irradiation, there's a very prominent induction of most of the SAS specific, the inflammatory genes. And then when you put WSTF back, so the inflammatory response of the senescent cells are largely repressed. And then we can also show at the genome-wide scale that the WSTF overexpression specifically affect the senescent, the SAS phenotype, the inflammatory response, and it doesn't affect anything else. So now this is overexpression of WSTF. So we can also do the reverse by knocking down WSTF. And this is what you see. We can stably knock down the WSTF. And then again, it doesn't really affect lamina P1, P16, a loss of phospho-RB. But then you look at the inflammatory genes, expression L6 and L8, you see when you don't have WSTF, these genes are massively turned on at a much faster speed. And so overall indicating that WSTF is a repressor of the SAS program of senescent cells. Okay, so the question is, mechanistically, how does WSTF block the SAS program of senescence? So we have looked at all the existing mechanism of the SAS. For example, Judy Campise's lab have shown that you need a P38 MET kinase ATM, and as well as the mTOR pathway to regulate the SAS. We have also looked at the cytosolic DNA sensing cGAS-STING pathway and the P53 pathway, and turn out that WSTF doesn't affect anything of that. So the question is, how does WSTF block the SAS program? Now, our hypothesis is that WSTF blocks the chromatin accessibility as part of the ISY complex. So remember, I told you earlier that WSTF is part of the ISY chromatin complex that is repressive. Right? So now our model is that in the control cells, the chromatin is largely closed. Now in the senescent cells, if you want to turn on the inflammatory genes, you need to open the chromatin, and this will allow the transcription factor NF-κB to kick in to do its job to stimulate the expression of the inflammatory genes. And now you see if the cells are open in this stage, if they see ISY complex, right? So the ISY complex is going to shift the open chromatin back to the closed chromatin to repress your gene expression. And that what happens is that we think that autophagy specifically degrades the WSTF and the ISY complex. So you will shift the equilibrium more to the open chromatin, leading to the stable and sustained expression of the inflammatory genes. So to test this hypothesis, we are running ATAC-seq to look at the chromatin accessibility of senescent cells. Okay, so I'm going to give you one example of the L8 or CXCL8 genes. You see in the control cells, the chromatin accessibility is low over the L8 genes. And then when you induce senescence of the cell, you see the massive opening of the chromatin measured by the ATAC signal over the LA genes. And then when you put WSTF, and this chromatin opening is largely blocked. Right, so we can again do this at the genome-wide level to show pretty much all the inflammatory genes are affected, and then also WSTF specifically block the chromatin accessibility of inflammatory genes, and it doesn't block anything else. And so here, I'm going to skip some in vitro data before I talk about the in vivo data to quickly summarize what we think is happening at the mechanistic level. So we think normally WSTF, as part of the ISY chromatin remodeling complex, will block the chromatin opening. The other thing I didn't have time to show you is that the WSTF directly binds to P65 subunit of NF-κB and also block the acetylation, which is required for the activation of transcription of NF-κB target. So these two mechanisms serve as a potent inhibitor of inflammatory response. Now, in the context of the senescence, the GABA-RAB binding to WSTF is specifically enhanced and greatly elevated. So this will lead to the nucleus to cytosol trafficking of WSTF, leading to autophagic degradation. And so then you relieve the inhibition, and this is how you turn on the inflammatory process of senescence at the chromatin level. Now, in addition to senescence, we can also show WSTF is a very broad and potent inhibitor of NF-κB mediated signaling in the context of senescence cancer and infection. So we found this is a constitutive nuclear inhibitor of inflammation, and that's why nuclear autophagy wants to degrade this negative regulator to turn on the pro-inflammatory response of senescence. So, okay, so the last question is whether this process is also important in vivo models of senescence, whether you also see loss of WSTF in vivo senescence, whether this is functionally important for inflammatory response. So we use a model to stably express the activity oncogene NRAS V12 in the liver hepatocyte. So we clone the NRAS into the Sleeping Beauty transposon system. And we use a technique called hydrodynamic injection. This is large volume, high speed injection through the tailwind. So this will lead to the specific expression of the oncogenic NRAS in the hepatocyte. So this will lead to oncogene-induced senescence in the liver hepatocyte, and that will cause the inflammatory response through the cells, and then they recruit a lot of the immune cells, leading to immune-mediated clearance of the oncogenic hepatocyte. So this is a potent tumor suppressive mechanism mediated by senescence and mediated by the cells. So this model is very robust. So if you set up the injection in day 0, you start to see the peak of the inflammatory response at day 6. And then 6 days later you start to see the clearance mediated by the immune system, immune-mediated clearance. And if you wait for additional 6 months, you start to see the liver phenotype. Now, in the normal setting, I should say that because the red cells are all cleared off by the immune system, so you don't really have to tune. But as we have, others have shown that if you block the inflammatory response, for example, by knocking out cGAS or STING, you start to see the massive accumulation of liver tumor because you lose the immunosurveillance of the oncogene. So what we are doing is we are either expressing GFP as a control or to force the cells to express the WSTF together with the NRAS. Okay, so first thing first, whether we can see WSTF loss in this model. So this is the day 6 of the injection, and then you start to see the NRAS expression in the liver. And then what you also see is that when you see the NRAS, you also see loss of WSTF in the nucleus. Right? So when you see NRAS, you also see the loss of WSTF in the nucleus. So normally the hepatocyte expresses very nicely amount of the WSTF, but when you express NRAS, the enter senescence, you also see the loss of WSTF. So this gives us the justification to put WSTF back. And now we can ask the question, what happens to senescence? What happened to the cells? Right. Now, this is the day 6. As I mentioned, this is the peak of the inflammatory response. You see in the GFP group, you have the NRAS expression in the liver. And then we also stand for CD45 as an immune cell marker. So what you see is that there's a massive immune cell infiltrated to the liver. And then what they are trying to do is to clear the oncogenic cells. Right? So what happened is that the the NRAS expressing hepatocyte enter the oncogene-induced senescence. So this secrete a lot of the inflammatory cytokine, the so-called the SASP. So this will recruit the immune cells and activate them, and then leading to immune-mediated clearance of the oncogenic cells. Now, what happened on the contrary is that if you force the liver to express WSTF together with NRAS, you still see that these NRAS are being expressed in the liver, but what you don't see is the CD45 infiltrating immune cells, because there's no SASP, and then there's nothing to tell the immune system to come over, so then you don't really have the infiltration of CD45 positive immune cells. So this is day 6, peak of the inflammatory cell. Now, starting in day 12, you will see the clearance of these cells by the immune system. All right, so you see in the control GFP group, the NRAS cells are largely gone because there's still this massive CD45 positive immune cells forming clusters. They are trying very hard to clear the oncogenic cells. And then what you see in the WSTF overexpression group is a total different scenario. You still have a lot of oncogenic cells, and then you don't have the CD45 positive immune cells coming over because, again, you lose the SASP. Okay, so what happened to this liver? They have tumor, right? So you see in the control liver after 6 months, nobody have liver tumor because the immune system took care of it. Right? So this is a potent immune-mediated clearance model mediated by the SASP. Now, if you look at the WSTF overexpression group, as I showed you earlier, there's no immune cells being recruited because there's no SASP. 100% of these mice develop massive liver tumor because you lose the immunosurveillance of the oncogene. So again, showing you that the loss of WSTF is really important in mediating the SASP phenotype, and that in this particular model, and then this is also a potent tumor suppressive mechanism. So you force the cells to put back WSTF, and you lose the tumor suppression. Okay, so to summarize everything we talked about, so we think WSTF is a novel nuclear substrate of autophagy. So we have shown the lamin B1 is the first substrate of mammalian autophagy in the nucleus. So this is through an unbiased proteomics approach. We think we identify WSTF as a very important substrate of nuclear autophagy in senescence. And then functionally, loss of WSTF by nuclear autophagy is really important to turn on the SASP program. And then I've shown you, if you put it back, you block the senescence. You block the inflammatory phenotype, and that has a very detrimental role in terms of tumor suppression. And we also show that WSTF is a direct binding partner of NF-κB and block the NF-κB acetylation. And this is WSTF is a potent nuclear inhibitor of NF-κB mediated inflammation. So what we think autophagy, nuclear autophagy is doing is that we think this is intrinsically a cell stress response. So the cells will turn on the nuclear autophagy, coping with the cell stress, and this will degrade lamin B1 and WSTF together with other substrate that we are still characterizing. So this will lead to profound nuclear and chromatin changes. And this, depending on the nature of the substrate, for example, we have shown the loss of lamin B1 is very important in reinforcing the cell cycle arrest. And then degrading WSTF will trigger the inflammation. We think this is beneficial in that it's a potent tumor suppressive mechanism. But depending on context, if you look at the natural aging, this will also block the tissue regeneration and will also contribute to chronic inflammation. So this is consistent with this idea that something is good for the young can turn out to be bad for the old. So that's why we think could be a potential therapeutic targeting window that we block the autophagy degradation of the specific substrate to promote healthy aging. So this work is mostly done by Yuan, a postdoc member, a postdoc fellow in our lab, together with Angelique. And then we also acknowledge Wade Harper, who contributed the proteomic work, and Torgen Johansen's lab in Norway, who help us to characterize the autophagy mechanism. So the most important take-home message today I want to deliver is that we are recruiting postdoc fellows. So my colleague Jay Rajgopal and our lab is recruiting a postdoc fellow to work on senescence, aging, and infection, and the lung. So we are part of the NIH Senescence Network Symposium, the consortium, and we propose to use single-cell proteomic approach to study senescence and the lung. So if you are interested in epigenetic senescence, aging, and the lung, definitely consider us. Yeah, so I'm gonna stop here, and I'm happy to take any questions you may have.
All right, that was fantastic. Really beautiful work.
Thank you.
All right. I see lots of questions coming in, so I'll go ahead and get started from the Q&A here. So from James Haslam, he wants to know, what's the method you use to find interactors of LC3 specifically in the nucleus?
Right, so this is a very good question. So what we did in this case, we specifically fractionate the nucleus, and then we performed the ATG8 IP mass spec from there. And then we are actually doing the 6 isoforms, and then they show overlapping yet distinctive binding partners. So short answer is we fractionate the nucleus, and specifically looking at the nuclear interaction. I do think this is a lot of people have done the LC3 IP mass spec before, but they are looking at the whole cell or the cytosolic fraction. So we specifically fractionate the nucleus.
Great, thanks. Aaron Havas, did you want to unmute and ask your question?
Yeah, Dou, wonderful talk. I'm always excited to see your work.
Thank you.
So you did hone in on the NF-kappa B effect of whenever you target WSTF, but I think early and late senescence, right? So are you specifically just targeting the NF-kappa B like arm, or do you think you're affecting interferon as well?
Right. So we have not looked at the interferon response yet in our model, because we are characterizing early time point of the senescence. I believe the interferon response is a much later process, as reported by John Sedley, also by Vera, so that you need to turn on the line 1, which I also believe it activates the CGAS sting response. But we have not looked at the interferon response in our model.
But you can in your mouse model, right? You did take that out quite far. If you have earlier time points, you could easily look for like some of the IRS, yeah?
That's right. That's right. That's something we can certainly look at. For example, in the fibroblast model in vitro, we can wait for much longer to look at the interferon, and then we can also look at the mouse models, of course.
All right, great. So there's another question from the chat. This is about the relationship between senescence and I guess normal aging, they call it. So Kristen Geronimo, degradation of WSTF1 by nucleophagy is a mechanism associated with senescence. Can it also occur in normal aging?
Right. So the question is whether the WSTF, loss of WSTF is a process occurred during natural aging. So we have looked at the aged mouse compared to the young mouse. We have looked at the tissue by tissue, and that we do not see loss of WSTF in most of the tissues, right? So, but what we have seen is that WSTF is specifically lost in the senescent cells. So when you have the senescent cells in the aged tissue, you start to see loss of WSTF just in that senescent cell, but not in the broad tissue. So we are actually testing this idea. Loss of WSTF could be a potential marker for senescence, just like loss of lamin B1. You don't really see that in most of the tissues, but you specifically see loss of lamin B1 in senescent cells. But in a way, lamin B1 is also degraded by nuclear autophagy. We think the lamin B1 and the WSTF is probably shared in the same pathway.
Great. And while we're on the topic, there was another question about the LC3 and the lamin. Does LC3 also recognize progerin?
There are papers showing that in the progeria model that you start to see co-IP between LC3 and progeria. And I also know there are papers showing the rapamycin, when you activate autophagy, you facilitate the degradation of the progeria in the mouse models, and associated with the extended lifespan of the progeria model. So yeah, so short answer is yes. So this is not from our work, but from what I heard and read, I do see that LC3 can co-IP with progeria.
Great. So in terms of the chromatin accessibility work that you've been doing, is it known, this is an anonymous attendee, is it known what regulates the opening of the chromatin for SAS genes in the first place?
Right. We have not looked at that yet, because we are looking at the repression. Right? But I believe a lot of other people have looked at that from the histone modification level. You have acetylation as well as the methylation that contributes to the chromatin opening. So we have not looked at that. This is from other works, other people's work. But I also think it's equally possible that they are chromatin remodeling complex, for example, the Swiss-Smith complex, that can open up the chromatin. But what we have shown here is the ISY complex can close the chromatin. And that's why autophagy wants to degrade it.
Great. And so Sweta Sikder had a question also related to the chromatin remodeling data. She found your talk very interesting, and she's curious to know how much of the ISY chromatin remodeling activity is affected upon loss of WSTF alone.
So the question is whether you really need senescence or you simply disrupt WSTF, you start to see the chromatin opening phenotype. So what we have shown is that in terms of activating the transcription of inflammatory genes, you absolutely need the transcription factor, for example, NF-κB in this case. And that NF-κB in the normal setting is floating around in the cytosol only in response to inflammatory or the infectious signal does NF-κB translocate to the nucleus. So what we know is that you need WSTF to work together with NF-κB in order to turn on the transcription factor, to turn on the transcription the transcription of inflammatory genes. You simply knock down the WSTF, you don't really trigger the inflammatory response, but you lower the threshold, and then the cells are more sensitive to inflammatory cytokines. So as I mentioned, we do know that WSTF directly binds to the P65 subunit of NF-κB. Right? So we do think they need to work together to turn on the transcription response. And that's why autophagy wants to get rid of WSTF to boost the inflammatory response.
Okay, great. And then there was a second part to this question. So in your ATAC-seq data in the senescent cells, do you find that chromatin accessibility is sustainably affected only at the SAS gene? So I guess, is there a broader response as well, or broader effect genomically?
Right, so that the question was to ask, what is the process comparing proliferating versus senescence? Right, so what we have shown you, and what I mentioned to you is the senescence versus senescence stably expressed WSTF. So when we look at what is globally open in the senescent cells, right, so you do see there's a massive upregulation, massive opening over the inflammatory genes. So we have that data, but I forgot right now whether you do see other changes. But it wouldn't surprise me there was also a close of the cell cycle regions together with other gene regions. So we have uploaded the data to GEO, so everyone is more than overcome to look into that.
Yeah, that's great. I'm sure some people will follow up on that. It's a beautiful data set that you have. So one more. Carla Valdiviso, probably pronounced that wrong. It's about cause and effect. So do you think the loss of lamin B1 is triggering the activation of P16, or is it the other way around?
Right. So this is a complicated question regarding the relationship between lamin B1 cell cycle and P16. So what is already known is that, so first you do have loss of lamin B1 in senescence. Right? So when you put it back, you delay the senescence, you delay the cell cycle arrest, and then you also delay the P16, P21 response. So it's hard to say what happens first, what happens later. So in a way, this is kind of a chicken and egg question. So from our data set, we also know that when you put lamin B1 back, you delay the P16, P21 response. But as I said, this could be you block the cell cycle arrest. So that's why you block the P16, P21. So I see that the P16 is strongly regulated at the epigenetic level. So it's not going to surprise me that there's some crosstalk between lamin B1 and the P16. So one more thing I want to mention is that the loss of lamin B1 is really important for the senescence-associated heterochromatin foci, the STAPI foci in the center of the nucleus that are repressive. And so if you lose lamin B1, this is very important for the formation of the SAF heterochromatin foci. In the senescent cells, you can put the lamin B1 back, and then you see the heterochromatin foci will disappear. So the loss of lamin B1 is very important for the epigenetic reorganization in senescent cells. So it's very likely to directly or indirectly talk to P16.
All right. Great. Well, thank you again. I think we should probably move on.